Welcome back, everybody, for another deep dive. Today we're looking at some really cool research that you sent in.
Oh, yeah, this one is really interesting.
So it's an article called Telomerase, Reverse Transcriptase Gene Knock-in Unleashes Enhanced Longevity and Accelerated Damage Repair in Mice. And this was published in Aging Cell last year.
Right, in 2024.
Yeah, and the research was done by a team of scientists at the School of Pharmacy at Nanjing University of Chinese Medicine. And also at the Regenerative Medicine Institute at the University of Galway.
Yeah, they're both really well respected in this field.
And this study, it looks like they've made some serious progress in aging research.
You know, this is something that scientists have been fascinated by for decades.
Right.
This whole idea of using telomerase to kind of combat aging.
Yeah, and this study, it really looks like they've hit on something pretty unique.
Absolutely. I mean, this research is really important because it uses a very precise gene targeting method to create like a heritable change in the mice.
So it's not just a temporary thing.
Exactly. It's not like those older studies where they relied on things like viruses to kind of temporarily boost telomerase.
Yeah, I remember reading about those.
This is a much more targeted approach.
Okay, so before we get too deep into this study, I think it's worth revisiting telomeres for a second.
Sure.
Just to remind everyone what they are and why they're so important.
Yeah, it's a good foundation.
Yeah, so, back in biology class, I remember learning that telomeres are like these protective caps on the ends of our chromosomes.
Exactly, and they're crucial for protecting our DNA.
But they shorten as we age, right?
Unfortunately, yes. Every time a cell divides those telomeres, they get a little bit shorter.
And eventually they get so short that
the cells just can't divide properly anymore.
Right, and that's when we start to see the signs of aging.
It's like our instruction manual starts to fall apart.
Yeah, that's a good analogy. So that's where telomerase comes in, right?
Exactly. Telomerase is like the repair crew for our telomeres.
Okay.
It can actually add DNA back to the telomeres, essentially extending their lifespan.
So it's like giving our cells a little bit of a tune-up.
That's a good way to put it.
So in this study, the researchers were basically manipulating telomerase in these mice, and they created this special mouse model called TurkKai.
Yeah, which stands for Telomerase Reverse Transcriptase Knock-in.
Okay, so how is this TurkKai mouse model different from previous studies on telomerase?
Well, as I mentioned earlier, it's all about how they boosted the telomerase activity. In the past, researchers would temporarily increase the activity.
Right.
But in this study, they actually made a permanent heritable change in the mice.
Okay, so like a fundamental change to their genes.
Exactly. They used gene targeting to insert the mouse TERT gene.
TERT gene.
Which is the gene that actually codes for telomerase.
Oh, wow.
They inserted that directly into the mouse's DNA.
So instead of just giving them a temporary boost of telomerase, they basically rewrote their genetic code to produce more of it naturally.
Precisely. And to really make sure that TERT gene was active, they used a powerful promoter. Think of it like a volume knob that really cranks up the gene's activity.
So they ensured that it was constantly churning out that telomerase.
Exactly. They really went all out on this one.
Yeah, that's fascinating stuff. I'm already eager to dive into the results of this.
Oh, yeah, me too. The findings are truly remarkable.
Okay, well, let's take a quick break and then we'll come back and explore just how these genetic tweaks affected the turkey mice.
Sounds good.
Okay, so we got these turkey mice running around with their supercharged telomeres.
Right.
What did the researchers actually find?
Well, the results are pretty amazing.
Did they actually live longer?
Oh, yeah. Significantly longer.
Okay, how much longer are we talking?
On average, the turkey mice lived about 16.57% longer than the regular mice.
16%? That's a pretty big jump.
It is, and some of them even reached a maximum lifespan that was almost 28% longer.
Wow, almost 30%.
Yeah. It's really remarkable.
But, living longer isn't everything. Were they actually healthier?
That's the key question, right?
Yeah, like were they just kind of hobbling around for longer?
Well, that's what's so fascinating about this study. The turkey mice, they didn't just live longer, they seemed to age better, too.
Okay, how so?
They didn't show any increased risk of cancer or other age-related abnormalities under normal conditions.
That's huge.
Their blood biochemistry was normal. Their organs developed normally.
So they were like the poster children for healthy aging?
Pretty much.
But wait, I remember you mentioned something earlier about cats with cancer, something about lung tumors?
Oh, right. Yeah, there was one interesting finding. When they exposed the turkey mice to urethane.
Urethane.
It's a known carcinogen. They did seem to be more susceptible to developing lung tumors.
Oh, so it's not a complete shield against cancer then?
No, it's not. It's a good reminder that messing with telomerase is powerful stuff.
Yeah.
And we still need to be cautious. There could be unforeseen consequences.
Absolutely. More research is definitely needed, but let's not get bogged down in the negatives.
Okay.
The article also mentions some really cool findings about tissue regeneration in these mice.
Oh, yeah, that was really interesting.
Did they actually heal faster?
Much faster. They did these experiments with skin wounds.
Okay.
And the turkey mice healed significantly faster than the regular mice.
Wow, so like how much faster?
They had less inflammation and quicker formation of new skin, and even hair follicles.
Wait, they even grew their hair back faster?
Yeah, it's pretty amazing.
That is impressive. So how's telomerase actually speeding up this whole healing process?
Well, it seems to play a crucial role in collagen fiber remodeling.
Collagen fiber remodel.
Which is essential for healthy skin.
Okay.
The turkey mice had thicker and more organized collagen fibers in their wounds.
So it's like they had a better foundation for that new skin to grow on.
Exactly. And there's more. They also saw increased migration of skin fibroblasts in the turkey mice.
Skin fibroblasts, what are those?
Those are the cells that are responsible for building new tissue.
Okay.
And telomerase seemed to give them a boost. They could reach the wound site faster and start repairing the damage more quickly.
So it's like telomerase is like a foreman on a construction site.
Yeah, that's a good analogy.
Making sure everything runs smoothly and efficiently.
Exactly. And the research didn't stop at skin wounds. They also looked at colitis.
Colitis?
It's an inflammatory bowel disease.
Right.
It damages the colon. And they wanted to see if telomerase could help protect against that kind of damage.
Interesting. So how did the turkey mice fare?
They showed much greater resistance to the colitis.
Okay.
Their colons were less inflamed and damaged compared to the regular mice.
So telomerase might actually offer some protection against this disease.
It seems that way. It's really promising.
So these mice are living longer, healing faster, and potentially even resisting inflammatory diseases.
It's pretty incredible, right?
It's starting to sound like the fountain of youth.
It's definitely a big step forward in our understanding of aging and regeneration.
But before we get carried away with dreams of immortality,
Right.
I think it's time to take a closer look at how they actually created these turkey mice.
Yeah, the science behind it is just as fascinating as the results themselves.
It is a bit of a mystery to me. I mean, the article mentioned using ES cells, and that sounds incredibly complex.
It is an intricate process, but it's what makes this study so unique.
Yeah, ES cells, embryonic stem cells. That just sounds so high-tech.
It is. It's a pretty complex process, but it's really what sets this study apart from previous research on telomerase.
Okay, so walk me through this. How do they actually use these ES cells to create the turkey mice?
Well, you see, in the past, when scientists wanted to increase telomerase activity, they would usually use viruses to deliver the TERT gene into the cells.
Right, like using a virus as a delivery system.
Exactly. But that approach has some limitations. The effects are often temporary.
Okay.
And there's always a risk that the virus could cause problems, like integrating into the wrong part of the genome or triggering an immune response.
So it's kind of a risky approach.
It can be. This new method is much more precise.
Okay, so they're basically changing the mouse's DNA directly.
Exactly. They're using a technique called ES cell targeting.
ES cell targeting?
Which involves modifying embryonic stem cells.
Okay, and remind me, what are embryonic stem cells again?
Those are the cells that have the potential to develop into any type of cell in the body.
Oh, wow. So they're like the ultimate blank slate.
Exactly. So they take these ES cells and they insert the TERT gene into them.
Okay, but how do you even do that? How do you make such a precise change at the DNA level?
Well, they use a technique called homologous recombination.
Homologous recombination.
It's like a cellular cut-and-paste system.
Okay.
They create a special targeting vector.
A vector?
Think of it like a molecular delivery truck.
Okay.
And it carries the TERT gene along with some other genetic elements.
So this vector is basically guiding the TERT gene to the right spot in the DNA.
Exactly. The vector is designed to match the DNA sequence at a very specific location in the mouse genome.
Okay.
And that location is called the ROSA 26 locus.
ROSA 26 locus.
It's known for allowing stable integration of foreign genes without messing up other important genes.
So it's a safe spot to insert new genes.
Precisely. So they use this vector to deliver the TERT gene to the ROSA 26 locus in the ES cells.
Okay, that makes sense. But we're still talking about cells here. How do they actually create a mouse with these modified ES cells?
Right. So the next step is they inject these modified ES cells into early mouse embryos.
Embryos?
Called blastocysts.
Blastocysts.
And as those blastocysts develop, the modified ES cells contribute to different tissues and organs,
Okay.
creating what's called a chimeric mouse.
A chimeric mouse.
Meaning it has a mix of cells with and without the TERT gene.
So it's like a blend of the original and the modified cells.
Exactly. And here's where it gets really interesting. They then breed these chimeric mice with regular mice.
Okay.
And because that TERT gene was integrated into the ES cell's DNA, it gets passed down to some of the offspring.
So some of the baby mice inherit this super telomerase gene,
Exactly, and then they selectively breed those mice over multiple generations
Okay.
until they eventually have a line of mice where every single cell in their body has this extra copy of the TERT gene.
And those are the Turkey mice?
Precisely.
Wow, that is an amazing process. It's mind-blowing to think that they can actually create these mice with enhanced telomerase activity using such precise techniques.
It really is a testament to the power of genetic engineering.
And the results are pretty remarkable, as we've been discussing.
They really are. These TERT-K mice, they offer a glimpse into the potential of telomerase to influence lifespan, health span, even regenerative abilities.
It makes you wonder what the future holds for this kind of research.
Absolutely. If scientists can figure out how to safely harness the power of telomerase in humans,
Right?
it could have huge implications for treating age-related diseases and improving our health.
It's really exciting stuff, makes you think about the possibilities.
Definitely. This study is just the beginning. There's so much more to learn.
Well, this has been a fascinating deep dive into the world of telomeres and aging research.
I agree. It's been a pleasure.
And thanks to everyone for sending in this study. It's really opened up some interesting questions about the future of aging.
Absolutely. Who knows what amazing discoveries await us?
That's a great point. Thanks for joining us, and we'll see you next time for another deep dive.